Kāti kia ora tātou, ka nui te mihi atu kia koutou katoa. Tātou me ki, ko whakarau ika I runga I te kaupapa o tēnei rangi. Te wahanga tuarima o te wānanga e kia nei, ko te pōrangi me te awhero, the crazy and ambitious. Nā reira, tēnei te mihi atu kia koutou katoa. Ko riro māku hei whakatūwhera I tō tātou tēnei me ki tēnei wahanga. Nā reira, mānawa mai ai te putanga o te ariki, mānawa mai ai te putanga o te tauira. Ka eke ki rongo rupe, ka eke ki rangi tā huahua. Tēnei te whatu kei au, kei te kaunga tapu te Maori, tū te whiwhia nuku, tū te whiwhia rangi, kei te whiwhia ai waho, kei te rawi ai waho, purutia mai waho, tāwhia mai waho. Tēnā tō Maori ora ka whakapiki, tēnā tō Maori ora ka whakakake, ko te Maori o tēnei ariki, ko te Maori o tēnei tauira, ko te Maori kei runga, te Maori e rangi, Maori ka pū kei waho, kei te whai au, kei te ao mārama. Turu o whiti whakamaua kia tīna. Hui e tā iki e, kia ora tātou. Well, welcome everyone. Welcome to this 5th session of Crazy and Ambitious. I will now just introduce our panel, well, our chair and MC for the next few sessions. So Rob Phillips is our chair. Kia ora, Rob.
Kia ora everyone.
And Nick Waipara is our MC for the day. Kia ora, Nick.
Kia ora, ngā mihi koutou katoa, ata Māori e.
Kia ora, Nick. Okay, I think I've got the rather daunting responsibility of introducing our first speaker this morning. And I say it's daunting because he is a very well-known and influential mover and shaker in his own right. I have to say at a personal level, Haami was a mentor of mine, a very important mentor and significant role model for me early on in my career. So I have the honour of Haami Piripi as our next speaker. And just a little bit of background on Haami. Haami is the chairperson of Te Rarawa Iwi and is a member of the Regional and National Iwi Chairs Fora. He works alongside the National Network of Māori Scientists, Te Tira Whakamātaki, and reports through to that forum on biosecurity issues, works with that forum on biosecurity issues. He's involved in a vast array of conservation initiatives, in particular Kauri Dieback, which, as you know, Kauri has a significant presence throughout the Te Rarawa tribal territory. Haami is one of the key architects of the Korowai model for the redress of conservation treaty grievances in Te Hiku o Te Ika and chairs the newly established Te Oneroa Atohe Management Board. And one little fact that I wasn't aware of, Haami was awarded the New Zealand Order of Merit for services to Māori. And Haami will be talking this morning on an atua Māori approach. Tēnā koe, Haami. Kei a koe, hoa.
Kia ora, tēnā koe. Eno, piki mai taku manu, ka ke mai taku manu. Piki mai, ka ke mai ki te taha o te wainui, ki te taha o te wairoa, ki te whakakiki, ki te whakakaka. E tū mai rā, ko rupe he kai pōwhiri, he kai manaki mo te ahuatanga o ngā kōrero. Hara mai rā. Tēnā rā tātou katoa. Given that the theme of this session is about controlling, protecting, and curing, I thought it'd be good to perhaps give a dissertation which describes an atua Māori approach and provides a legitimacy of purpose, perhaps, for the carrying out of kaitiaki duties as a theological backdrop, I suppose, for conservation management. And there'll be a question, I suppose, about the provenance of that conservation knowledge that sits within the Māori world. And in our claim negotiations, we argued that there's no way that anybody could claim anything of us pre-1840 in terms of our roles and responsibilities around natural resources and the environment. So that pre-1840 provenance goes back well over a millennium, and that is an important era of this country's genealogical, chronological history. And there's all that time span of monitoring, of observations, of trials, of establishing a knowledge font has really provided Maori communities with the ability to discern medicines, cures from the forest, and based upon their knowledge, scientific, I would say, of what the properties of those plants and that were, and established a whole echelon, if you like, of Rongoa Maori, which has diminished over time, but is now beginning to make a bit of a re-emergence. And with it is the indigenous nature of the knowledge and the nomenclature that's attached to it. So the Atua Māori approach relies on a Māori worldview as an indigenous paradigm, and it's a unique and independent explanation of the universe. It's a paradigm that has a genealogical methodology for defining and demarcating relationships, resulting in a connectedness between all things, including their placement and context, and in relation to other things around them. Thus, it can be said that in the Māori world, if you are not a part of the whakapapa, then you shouldn't exist. And that model of acknowledgement is applied equally to the entire spectrum of Māori existence, I think, including our astronomical understanding, culminating in the planet that we know as Papatuanuku, and the space around it that we know as Ranginui. These are the two primordial entities, of course, which parent all life on the planet. And the progeny of these entities emerge as elemental phenomena, conceived before their separation, but becoming manifest within a cosmic pantheon of what can be described as siblings, I suppose. And these are who we know as Atua Māori, each possessing their own features and attributes, and subsequently their provenance as entities becomes expressed as characteristics among a hierarchy of deities. The practice of Atua Māori tikanga is drawn from an epistemology of these entities and the theology which shapes them and describes their functions. This notion of genealogical determination enables form and function in respect of the Atua deities and the relationships they have with everything else that exists between sky and earth. This includes human beings, of course, Maori ones at least, who have been, according to our wānanga or school of learning, created by the deity Taine Nui Arangi, and then undergoing a process of Taine Nui Arangi procreating with us to provide humankind with a line of descent that has two threads of two properties, perhaps, one divine and one material, or seculum even, but it's the divine and material aspect intertwining that establishes the basis and rationale for our role as kaitiaki. It connects us in terms of whakapapa with our cosmic surroundings and all natural phenomena between sky and earth. So consequently, what is known as Te Waunui o Tāne is an integral core biodiversity that interacts with others and with the domains of other deities. Together and intertwined, this is what we know as Te Taiao. The domains of the deities can be observed as elemental phenomena. For example, Tawhiri Mātea could be described as the atmosphere, or Papatuanuku, the planet, but it would be incorrect to consider Te Ao Māori as a personification of the elements. Although they are in fact ancestors in one form or another in a multi-dimensional relationship, in hierarchical terms we are affiliates of the deities by virtue of our birth, who cannot logically possess or even appreciate or comprehend possibly all the attributes retained by Atua, and certainly could never place ourselves at the center and consider we possess the be-all and end-all of intelligence on the planet, let alone the universe. And that is how we as Māori view the Kauri, the oldest, largest, and most far-reaching functional canopy of Te Waunui o Tāne. So I want to focus a little bit upon Te Reo o Te Kauri, because I've become so concerned that we haven't yet found the answer to solve the problem of kauri dieback. I want to begin by sort of dismissing the assumption that trees can't talk. That was the sort of thing that European explorers would think about us as indigenous people, like non-humans whose noises made between each other could never be described as a language. That's a very old perspective, I know, but it's the same sort of thing. And I think we are missing the ability to capture, to understand, comprehend the reo o te kauri o ngā rākau. And I'm suggesting that we also, perhaps at our great peril, have failed to hear the language of Te Waunui, or Tāne, of which we are an integral part. The ancient karakia of our people does take cognizance of it because communication is required in order to achieve any effect. But there is another wānanga, and I'm talking about here the avenues of hearing, somewhat like a view shaft within which the kauri can and does express itself. Let's start with the ngata, the kauri snail, which at a certain time of the year scales the outside of the tree and turns its back to a prevailing wind. The whistle that occurs as a result of that snail back being turned in a certain way is barely discernible to the human ear. However, we know from our cultural knowledge fonts that the sound that is emitted by a certain wind upon the shell of the snail is in fact a mating call, facilitating the future survival of the species, and could be described as an aspect of the voice of the kauri. So it could be thought of in a way that it is clear that there will be many other aspects of that voice of the kauri. A culmination of these aspects could result in an enormous voice, seemingly unheard by us as humans. But there's one aspect of a te reo that cannot be denied, and that is the voice expressed by its destruction. It is an intensely moving experience at a spiritual and emotional level to witness the build-up to the crescendo and the falling of a mature kauri tree, as it is taken down within the forest, as the greatest canopy that exists, where it provides literal protection for 16 other species, flora and fauna. The reaction of those life forms alone are a catalyst for concern, and then there are the birds, other progeny of Tāne, who during the process of felling the rākau rise and protest in alarm to an incredible crescendo until the point of the tree actually falling. And then as the tree falls, as each branch hits the ground and then it creates its own crushing sound, it creates a sort of a rhythm of dying, I suppose. And then when the tree hits the ground, the impact can be heard from 8 kilometers away, and everything goes dead silent for a long time before it gradually starts to come back to life. And so in this scenario, it's difficult to conceive of a situation where the children of Tāne could not communicate with each other. And life is not like that. And taking a biodiversity lens, we can see the similarities with the notion of whakapapa and the concept of kaitiakitanga. So when science catches up to indigenous knowledge and when the value of science is utilized by indigenous people, we may achieve that holistic approach I think that we need. 2 arms will always be better than 1. And in every context, Maori knowledge has actually been excluded from the empirical nature of research in the past, enabling ethnocentrism to extend into the educational and training institutions and contributing further to our marginalization from our ability to grow and develop our kaitiaki roles and responsibilities with the added rigor of scientific pursuit. So now it's only now that the value of it is being realized as cultural capital and utilizes a growing awareness of its importance, shows that a lot of projects would not be able to succeed without it. And associated with that development are the new arenas of research, utilizing high new technology, ICT, like 5G, for example. And as we begin to explore the opportunities in a thing like 5G, we can see that there are avenues that we can jointly pursue. For example, there's work underway to understand how plant life, or more particularly tree species, can and do communicate with each other. And they've been able to, these people have been able to record the sounds that are emitted that they believe are the tones of communication. And imagine what it would be like if we could employ nanotechnology to be able to explore that notion further and to be able to address pathogens like Kauri dieback or myrtle rust, for example. We really need that capacity, I think. And so in the death of the Kauri, we hear its voice. But even after the tragedy of its death, the Kauri is still speaking in the work of the wood and the production of taonga from the timber. And the Kauri in Tangonga is 30,000 years old, and we're really privileged to have access to that particular voice of the Kauri, ancient voice of the Kauri. So coming back to the issue of control, protection, cure, when it comes to legitimacy of purpose, what I'm saying here is that as Kaitiaki Māori, our legitimacy of purpose is drawn from our genealogical relationship with the taiao, with the environment, and with the atua Māori. That are the deities of the domains within it. And from that legitimacy, we gain our ability to practice a sacred role, really, of kaitiakitanga, based on that relationship and based on that affiliation, but also based on our ongoing association. And this is what I want to really finish on, and that is the incredible loss of opportunity in not utilizing the ranks of kaitiaki nationwide. In fact, we have probably historically repressed them, the ranks of kaitiaki, first by turning the land from forest agriculture, and then secondly by the agricultural usage, excluding person usage, and creating 2, 3, 4 generations in some places of young Maori people who have not had the opportunity to access and participate in their kaitiakitanga roles and responsibilities associated with the natural resources in their rohe. And that is a really untapped resource. Almost too late, because we're going to have to go back and relearn some of that stuff, I think, some of our young people. But maybe we've had plenty of some exercise to help us along that way. I'm not putting down new technology. I think we need new technology, and the whole idea of succeeding in an initiative like being predator free will depend on innovation and motivation and participation. And I've said on the arena around predator free, it cannot be done without kaitiaki Māori. We just wouldn't have the army to do it. And that's where the kaitiakitanga principle, the hapu associated with them, can be engaged and utilized in a much more comprehensive and, I think, useful conservation management regime. So I think I've used my 20 minutes, and I'll leave it at that.
Kia ora, Haami. I mean, that. Thanks for your kōrero today, as ngā mihi. That's given a great start on our journey for the Cody Land Summit. So it would be a virtual homai te pakipaki for the audience if we were kanohi ke te kanohi in a whare somewhere. But it's my role now to take on the Q&A. But before doing that, we're going to introduce Rob Phillips here, who's going to chair the rest of the session. Kia ora, Rob.
Kia ora.
Welcome. No mai haere mai. So you've
Thank you,
had
Nick.
a long history with the Biological Heritage Challenge. So kia ora for joining us today. And we're going to have a great panel of speakers following. But further, we're going to have the Q&A for Matua Haami. So there's the great ngahere of Warawa, and some of the most magnificent rākau I've ever had the privilege to see and enjoy being within. So in your rohe, it's a major tragedy how our kauri in Aotearoa has been afflicted with many events over the last 200 years. And I guess the last being kauri dieback and climate change and all that's upon it. The knowledge that we're seeking, the mātauranga and the kaitiakitanga, I think that will take us, ourselves, and our ngahere beyond. Do you think we're in a place to do that yet, or do you think we are still barriers for doing that? You know, the system, the approach, biosecurity management or long-term pest management or those sort of words. We haven't had the rangatiratanga or the leadership bestowed. Does that matter, do you think, in facing kauri dieback?
Yes, I think it does. It comes down to having the ability to do something about it, and then having the ability to provide to blaze a new trail in terms of a knowledge font around it. And the two need to work together to produce the chemical reaction. We need to find the solution. And I think that as Maori communities ourselves are gaining more ground in the educational institutional arena, it's getting better. But I think it also can come down to time and place and circumstances. And right now we have got a Minister of Conservation who I think is awesome, and is prepared to make to make changes to make it right. And because if we were to not take this approach, it would lead to a continued marginalisation, and so partnership is the only way to really go. We can't be just a stakeholder. I think people underestimate and misconstrue perhaps our role. I think we have got a public good role. I remember way back when I was early conservation years, the conservation groups used to call us private landowners, and Maori only one of the things is private landowners. But actually, that's not true. We want to conserve and preserve areas for everybody's sake, and as well as the sake of the taonga. So I don't think I think we really are a partner, and as an Indigenous partner, we have great value.
Kia the terms that Māori have been subjected to, like stakeholder, end user, I find a particularly pointed one. But the I think the kaupapa of Ngā Rākau Takitaki and what we're doing now in the community of knowledge and research and recognition of mātauranga and leadership in both the protection but also the knowledge that's going to save our rākau. I think, do you see that as a strength from a divided research science system that didn't recognise, and some institutes recognising mātauranga and all that comes with mātauranga, mohiotanga and kaitiakitanga? Do you feel that we're on the journey now to solving some of these issues?
Yeah, I think we are. We certainly are in terms of process. In terms of outcome, perhaps it might be a little different. I'm not too sure, but certainly are in terms of process. But the point I made before is that a millennium of observation, of trial and error, is really scientific expression, and the knowledge that comes from that is actually a science in its own right, and it's often been considered just be mumbo jumbo, or some kind of incantation to I don't know what for, but incantations have a purpose, and that purpose is to connect with the reo of Te Wānui, or whichever domain you're working in, and that's what science is missing. So I remember when we first got involved in shifting tuatara from Takapōreo, Stevens Island, to other islands. It was very difficult at first, but I observed that once we introduced the notion of karakia, proper karakia and ritual and process to the facilitation of the exercise, it seemed to go a heck of a lot better. And I think I've seen it myself with my own eyes, the importance of karakia to make things work.
But, that's certainly so, and I will pass an opportunity for Rob to maybe comment or say something as well.
Yeah, Kira, thanks, Nick. Kapai. There's a question from Desi just in the chat, in the question thing, so I'll start with that one. And Desi asks, Haami, do you think that the green paper will open the door for an opportunity to break through in terms of next steps?
Yeah, I think it will. But one of the things that we have to really try and avoid repeating is underselling the historical aspects of how we got here. And it's easy to just say, "Oh, we'll cut that off and we'll move on from here. We're moving on," and it's all good to move on, but in the process of moving on, we have to fix what's been behind us. So I think there's definitely great potential there as we look ahead, but we can't forget our past as we do that, and that past drives us to certain behaviors or certain institutional arrangements for our future. So Nick was talking about those terms about being end user and stakeholder. Well, the one I'm most afraid of is volunteer, because my people stand on the veranda and watch all these shiny ass conservationists go past, and they're wondering why they're not involved. And so it's something we have to just bear in mind, I think, in our thinking.
Sure, that's good. That's good. There's another couple of questions, I think, here too, Nick. I just can't quite pick them up. Can you see them?
I can't see them either.
They're in the live
flooding
Q&A
in,
chat.
Haami, but we can't pick them up.
In the live Q&A chat, isn't it? Yeah. So in a similar vein, Haami, so a lot of work going on now to operationalising and getting into a national type strategy. Do you think the Mātauranga Maori's embedded well enough into that plan and process? In terms of the operational, what are we doing about things? Is that all integrated together, or is it still not quite where it needs to be?
No, I think it's still fairly fragmented, and what happens is that various government departments or agencies, they charge off, they find a tauhanga somewhere and they charge off on their mission, and they develop this thing for themselves, and it's not really aligned with a wider lens of understanding connectivity, I don't think. And so, and as a result of that, I'm working with the Y262 issues as well. As a result of that, agencies progress new bits of legislation. I mean, the conservation is doing it right now with the Wildlife Act and stewardship lands there. So just changing the goalpost a little bit and disabling us from taking full advantage of the situation. So it's like any relationship, it's got to be constantly worked at. As someone explained to me once, the difference between happiness and contented. Being happy and content. When you want to be happy, you got to work every day and make sure you get there. But being contented, you can plan for that, for everything, get everything ticking over in a much more predictive way. And I think that's what we have to work towards, is a contented relationship, because it is a relationship in a very similar way to any partnership between two people or two groups, whatever it may be. It's a partnership that needs to be honoured. And there's a lot of dishonouring going on in the past, and there's been a lot of lists kept happening with our waka, but it's only a list. So while it's only a list, there's hope for us to bring it back to an even keel, and I think we've shown that really during the COVID response, we're capable of creating that even keel, and in that partnership. And so I think there's definitely it's the only way for us to go, really. It's a potential that's what they call into Maori, Maori te maungaro, the thing that has been lost, the thing that's missing, the missing element, and te maungaro, and I firmly believe that's what that is.
Cool.
Well, kia ora again, Haami. This is, as I said, has put us on a firm footing for today's kōrero and whakaaro, having your whakaaro and your here as our guiding light for all things Cody and Cody Summit. So kia ora. Thank you.
Kia ora. Tēnā koutou.
Ngā mihi. So we've got a number of ngā mihi to all the comments we've just seen. Jan, Rebecca Fuller, ngā mihi. Kate Nolan, ngā mihi. Sorry we couldn't get to everything, but we're moving right along. This is an exciting adventure with the Control, Protect, Cure research theme. So ngā mihi kōrua Dave Milner and Marianne Wood, who are the co-leads for that research theme. So acknowledging which theme we are today, first up, is a very good thing. So kia ora to you both. And also we are now going to see what that whole theme is about. So I'm hoping the producer has the video on play, because we're going to pass through that now. Kia ora.
I'm David Williams, and I'm a chemistry professor at Auckland University. My professional career has been around basically how to measure stuff, kind of sensors and things like that, chemical measurements. So there's a group of us at Auckland. I work a lot with Yadranka Travis-Sadich, so she's another professor in Auckland, and we work on measuring things. And Marion approached Yadranka first and said, there's this problem about trying to measure the greebies in the soil that are going to go and infect the kauri trees. How can we detect them? Yadranka asked me, and we have a bit of a brainstorm and nut it out, and said, well, maybe we could do it, and this might be a way to do it, and so write a proposal. And they said, looks like it might work. Well, it's got a long name.
What's the long name?
I can't. I think it's called a microcytometer. I think that's something like that. Or a microfluidic flow cytometer. So what it is, right? So I haven't got one. So you have to try to imagine it, a little tiny block of plastic. And in the block of plastic is a channel. and the channel is smaller in dimension than a human hair. So it has to be just a bit bigger than these little beasties that you're trying to measure. The idea is to persuade the beasties to travel up the channel, right, to swim up the channel. And if they swim up the channel, they provide a little bit of a blockage. They don't block the channel, but they're kind of a little bit of an obstruction. And if you have a pair of electrodes in the channel, and the beastie goes past the electrodes, you can look at the way electric current flows between the electrodes, and it detects the little blockage. So you get a pulse as the beastie flows through the gap between the electrodes. So the channel is smaller than the dimensions of a human hair, and so are the electrodes, and so is the gap between them, because you're measuring something small. The first level is are you smoking dope, right? Is this just really stupid? Can you actually measure something? So Piripi made these things, and sure enough, you can see little polystyrene balls going down. But polystyrene balls are not beasties, right? They're hard, and beasties are soft. And so Shamini worked out how to grow some beasties, which is non-trivial. And then Piripi showed that actually you could see them. So that's kind of cool. So that's the stage we're at, basically. Yeah, got a signal. Amazing. Well, first you've got to measure something, so you've got to get a signal, right? So you've got to figure out the electronics required to get a signal. How big a voltage have you got to apply, and what frequency should it be at? It's an alternating one, so it's doing this, and so how fast should it go? And then how are you going to detect that? So the next part of it, the project was actually the detector. Can you detect this thing? And Logan, we were sort of having discussion, and we found this thing that you could buy, an actual chip that actually was just right, and so they got hold of it and they were able to put it together, and it works. Now you've got the most difficult part, which is you can detect these things if they go past the electrodes. So how are they going to get there? So now we have to do experiments with these little beasties, actually with a version of the beastie that isn't infectious to trees. So my understanding of this is that there's the chemicals that are coming out from the roots of the kauri trees, and these things detect those chemicals and swim towards them, and then get into the root and go up the root, right? Okay, so it turns out that carrot juice apparently works to attract these things. So now we can now sort of start to explore what happens to these things when they've got when they detect some carrot juice, right? How fast do they swim, and how far do they swim? And can you get the carrot juice into this tiny channel in such a way that it seeps out the end just enough to attract the beasties? And when the beasties come along, will they go into the tube? Will they actually go into your channel, or will they just stick at the outside? So do we have to make a shape at the beginning of it? And how fast will they swim? Will they stop swimming? Will they stick to the walls? And all this kind of stuff. And when we can do all this observational thing, right, about the way in which these creatures are behaving, then we'll know how we shape the inlet to the channel, where we put the carrot juice, how we get the carrot juice into the channel and just kind of just seeping out the end. What's the concentration of carrot juice, right? You don't know. And where do we put the electrodes? So there's a tree, somewhere around there are beasts. You want to know if the beasts are getting towards the tree, where are you going to put the detectors? Because if the beasts go, the detector's here and the beast's over there somewhere, the detector's useless, right? So there's a whole other thing about where do you put these things and how many of them do you need. The sense is only the very first part of all of this kind of thing. You've got to have something which is monitoring the signal automatically, looking for pulses. And it has to say what's a real pulse and what's a fake one. To me, it was like if I was asked if I'd like to, contribute in this, and you go, oh, of course, right? If you can find a way to help stop this stuff. Well, you're providing new ways of seeing, I think. Ways of seeing things that you can't see with your eyes. a lot of other stuff that I do is do with measuring air quality, right? Measuring gases in the air. We can't see it, you can't smell it, you can't taste it. And the company I work, they have a slogan called making the invisible visible. And a lot of what I do is like that. It's measurement measurement like detecting these little beasties or, chemicals one sort or another. You have to extend your senses. And it makes this thing visible. And when you've done that, now you can integrate it into all sorts of things, right? It becomes now part of your knowledge of the whole system, and what are you going to do about it?
Ko Waio, ko Kirireihana tōku ingoa, he uri no Ngāpuhi me te Rarawa e tētaha tōku pāpā, he uri no te whakatohea ngai tūhoi e tētaha tōku māma. Dave I met because he used to work for Ngāti Rangi, and the basis of the cultural framework that we'd done the work from comes from the Ngāti Rangi work I did. So with Ngāti Rangi we did a forest health or Ngāhere Ora framework to look at what the health of the forest looks like from their perspective. So Dave knew I'd done that work and asked me if I'd be interested in doing a similar kind of thing in this research, which is now being coined cultural monitoring by quite a few people. So
Is that what you call it?
I would say a mātauranga monitoring. It's framing the environment from their own perspective and understanding it from their own perspective, capturing information from their own perspective, and then being able to communicate that back out into reports, board meetings, but ultimately it's information to go into decision-making forum where they can start making decisions with mātauranga, Māori knowledge, as well as other knowledge systems as well. So at least we're participating in that co-management space from our own point of view. What I've observed from doing this kind of monitoring, that's what iwi are really wanting, is their own data so they can present it in their own way and to contribute to decision making around their own resources. We start with noho taio or marae noho, where we extract out things that are the values of hapu and iwi, whakatauki, because each iwi obviously has their own mita as well, so their own dialects, their own worldviews. So we try and capture that. Then from that, we'll turn that into some kind of metric matrix, where we can use it to measure those intangible and tangible concepts like whanaungatanga, manaakitanga, Maori, from their own point of view. Then we turn that into a field sheet. We test it in the field with their kaimahi on the ground so their kaimahi can feedback. So it's a reiterative process. It'll start off with their elders, it'll go through to their people on the ground, but we take it back to the elders, the kaumātua, or the senior specialists that they identify in their iwi or hapu, and just to get them to okay it, to mandate it, so that we know that we've done the right process to ensure that this is a true reflection of the iwi or hapu we're working with. We've moved into this post-treaty settlements, a sort of phase where Māori now have a say in our natural environment and our resources, and so they want to, but they want to present their worldview on it, their perspective on it, and their data on it as well. We haven't been at the table, at the decision-making table, for a very long time, and I would say that this kind of, pushing towards getting this has been, got traction in the 80s, probably started in the 50s and 60s with our tupuna, wanting to not just get their land back, but also having say on the land that they didn't get back, like national park land. Or the mataitai they call it, or the marine reserve land that they've got now and the different names. They had those coming through legislation, but enacting that on the ground and affecting it in the decision-making co-development spaces, there hasn't been a lot of traction we've gotten with that. So I think it's coming to a point now where we're starting to get traction. We're starting to understand the research system and how to collect knowledge in that particular way so we can do it our own way, but from our own worldview. Because unless we're making that those decisions informed from our own worldview, it's not representing our worldview either. How we've broken the framework down, or how Ngāti Rangi broke it down, they broke it down into what they call domains or attributes, which are areas that we wanted to focus on to look at the health. So for Ngāti Rangi, it was rongoa, wai, tangata, rākau, and the ngahere in general. So we've gone with the same things. We've also added in for this particular one is awa. I'm looking at the awa itself. So there was two sort of sections on water in the forest. When we did the Ngāti Rangi framework, they concentrated on what the wai was in the forest. That was measured by a metric of Maori, how flourishing the Maori was in the forest based on the water content. So when you're in the ngahere, there's a smell. It's usually the decomposition smell of the earth. So when it's really moist, they were measuring that as a real healthy smell in the forest. And then when it's dry, it's not as healthy. When we were in the bush talking about that yesterday, because there's been droughting up here, it was a real dry smell, even though it's this late in the year and the forest's up there. So it's understanding those things that happen in the forest, the processes that happen in the forest that make the forest well and healthy, and water being a big part of that. In this particular in this research stream, we've used also looking at the awa, or the lakes that are associated with the forest that we're working into, and measuring the health of that through the presence of taonga species in those awa, the clarity of water, how clean it is, how not clean it is, depending on where the source is coming from too. So we've added that other dimension to this particular one. What we still have to develop is cultural indicators or indices around the Kauri dieback and the myrtle rust. I know Ngāti Rehi has done a bit of work in that space. We looked at the Kauri health indice that Tui Shortland and Julia Cheetham have done. A lot of what we've got into ours marries up with that one. So there's some specifics we're still wanting to find out. So having kōrero with people like Tohe Ashby would be really valuable to refining what that might look like for these kaitiaki and kaimahi. That connection to whenua, I think is just so important for so many reasons, physically, mentally, spiritually. We are much happier beings when we're connected to the whenua. And whenua, moana, awa, the taiao, whatever that looks like for anybody, humans are much happier in that way. And I'm a conduit of what is needed at that time. A friend of mine, Hema Wihong, she always comments that we're just the baton holders, the rākau holders of this moment, because our knowledge is intergenerational. And our work is intergenerational too. It started with our nannies and aunties and doing all the mahi they saw that was important of that time. And we're just the conduit of this time as far as I'm concerned, yeah. Some people are special, and have those special abilities and hold those special mātaurangas, like I mentioned before, Rereata or Tohe or Palmer Gowan. But mātauranga is not supposed to be held and kept, it's supposed to be shared, so it has that ebb and flow like a river, so it moves through the generations and it picks up the gold bits on the way, the nutrients on the way when you're in a river, until it deposits it at another place. So I guess I see mātauranga like that, being in the environment, it reflects, our knowledge reflects the environment of the time.
Rarawa. Control, protect, cure. There you've got it. So our chair, welcome, Ngā mai haere mai back. You've had a long journey with the challenge, haven't you? A member of the co-governance Rōpū for Bioheritage since its inception and co-chair for the last 12 months. So you've got a wonderful perspective in this whole session as our chair. So I'm going to hand it over to Rob to continue the kōrero for this. Kia ora.
Kia ora. It's a real privilege to be part of this challenge and doing a bit of the mahi to do this important work. But look, before I introduce the next speaker, can I just further talk about the control, protect and cure, and the need for, and you can see it's really clear to everyone, I'm sure, that's zoomed in on this session around we desperately need a suite of fully integrated management techniques and tools if we're to save our precious Nahiri. The control, protect and cure team have been using an extensive outreach process to develop and socialise new technologies, assess important values and concerns, and ensure research operations and case studies integrated effectively. This diverse team has been brought together to develop and explore strategies and tools that can be used in the fight against these devastating organisms, and so it's so important. So look, can I introduce the next speaker, who's Eiren Sweetman. She's an important. She's got a PhD from Otago University in biochemistry. She's passionate about conservation and environmental issues, and she's moved into the research world at Victoria University, working with Dr. Monique Girth's research team focused on finding solutions to kauri dieback. So I'll hand over to Eiren now. Over to you, Eiren. Thank you.
Kia ora koutou, and thank you for this opportunity to share our group's research on the umaiceae Phytophthora agathodicida, the pathogen that causes kauri dieback disease. So as the little introduction said, I completed my university studies in 2018 to become a molecular biologist, and I'm a lifelong conservationist. I joined Dr. Monica Girth's research group in 2020 University of Wellington, and this is a recent photo of our large research group with a few past but mostly all present members. Our kauri dieback research is based in molecular biology and collaboration with Mātauranga Maori knowledge holders. And importantly, we've been able to build upon our collaborations and relationships with mana whenua in this area by being a part of Te Whakahonanga, developed by Ngā Rākau Takitaki co-leads Dr. Marion Wood and Dave Milner. So the goal of is to discover safer and more effective treatment or curative options for kauri dieback. And we have developed specialized screening techniques with which we can screen compounds, extracts, and even other microscopic organisms such as fungi and bacteria for their ability to inhibit the key life stages of Phytophthora agathodicida. And I have these life stages illustrated here. We have the oospores, which are introduced into an area of kauri growth, typically by human activity, but also animal movement. They germinate to form sporangia, which are these structures here that then produce zoospores. And zoospores are the infectious life stage. They have these tails, which they can use to swim through water in the soil towards kauri tree roots, where they attach to the outside, encyst, and germinate again to produce mycelia. And this mycelia spreads through the whole root system of the kauri and eventually stops the transfer of water and nutrients to the canopy. And this is just a little video of those 3 key life stages that we look at. This is an oospore, and there's another one further back. Surrounded by mycelia, and swimming all around the screen are little zoospores, and you can't quite see their tails, but they're there. So I'll now briefly describe our screening techniques that we use and show some of the success we've had in finding potentially effective treatments. Our first screening technique is a mycelial growth inhibition assay. So this is where we grow Phytophthora agathodicida on an agar media present. And you can see in this image on the right that mycelial growth has stopped after only about a centimetre of growth out from a central Phytophthora plug, unlike this image on the left where there was no treatment present. We screen the mobile and effective zoospore life stage by testing whether substances can stop zoospore movement and also whether they can also inhibit their encystment and germination once they've stopped. So on the right are zoospores that stopped immediately to a treatment, and they've also failed to germinate. And on the left, you can see that these zoospores have started to germinate. You can see little mycelial strands emerging from the encysted zoospores. So we published a paper in 2019 on flavonoids we identified from Kanuka extracts that we were able to show had anti-Phytophthora activity. And we used those two screening techniques that I've described. But back then, we didn't have a good method to look at the effect of these Rūnanga extracts on oospores, which are the hardest life stage to treat, as they're essentially survival spores designed to withstand all environmental challenges from extreme heat to cold to crushing to movement and more. We now have two assays we can use to tackle oospores. We have a germination assay, which allows us to see whether oospores will germinate in the presence of a treatment. And we have a viability assay, which allows us to see whether a treatment will kill oospores completely. So for our viability assay, we use a combination of two dyes, a green dye to detect live cells and a red dye to detect dead cells. So the green dye moves freely into oospore cells, and if the oospore is alive and metabolically active, it will cause the dye to glow green. The red dye, conversely, cannot enter oospores unless their outer cell walls are compromised, which happens when a cell dies, and it glows red when it binds to nucleic acids. So we can treat our oospores, then incubate them with our two dyes, and then look at them under a microscope and take a photo of all the oospores present in that field of view, then an image in the green channel and an image in the red channel, and then we will count the green cells as viable, alive, the red cells as non-viable, dead, and the yellow cells as weakened, because we assume they're both metabolically active, but they also must have compromised cell walls. And in this way, we can end up with an estimate of viability rates in a population of oospores after treatment. So a key aspect of our research focuses on exploring native Rūnanga plants for their potential to suppress the growth and spread of kauri dieback using all of our screening techniques. These native plants have been selected after collaboration with Mātauranga Maori of Ngāpuhi, but we need to progress our initial findings through leaf and plant trials before they're proven useful for use out in the field. But an exciting follow-on based on some of our findings is a long-term trial we have underway using aligned funding to test companion planting kauri with select Rūnanga trees as a protective measure and to stop those trees from becoming infected. Thank you for your attention. Kia ora.
Kia ora. Many thanks, Maureen. It was impressive. We'll have a chance to ask some questions a wee bit later, but the next speaker we've got is Grant Smith, Dr. Grant Smith. So Grant's first experience with myrtle rust was in 2013 when he was based in Canberra with the Biosecurity Cooperative Research Centre. And since the 2017 incursion into Aotearoa, Grant has been leading a number of projects and programs to understand the impact on our native flora, and also contributing to the important work around sequencing the fungal genome. Now, this is sort of high-tech stuff, which is actually a fundamental science which we need to have if we're going to make progress. So great stuff. He's currently leading research to understand the fungal pathogen itself and manuka and plant resistance in the Manuka. So can I hand over to you, Grant?
Sure. So thanks for the introduction, Rob. Tēnā tātou katoa. So I'm going to talk a little bit about identifying the cause of myrtle rust, and I've framed this presentation up around why we actually need a diagnostic for myrtle rust in the New Zealand context. This is a collaborative project between New Caledonia, Scion, and Plant Food. And so diagnostics is one of the 5 core components of the biosecurity continuum. Often response is driven by the diagnostic result, and it's subject, like all the other components in the continuum, to feedback. Now, diagnostics is about identification, and it has to be specific. You only want to detect the target of interest. You do not want to detect. And it also has to have a known level of sensitivity. How many propagules, how many units of that organism that you're seeking to find do you know that you can detect? Now, diagnostics is subject to both type 1 false positives and type 2 false negative errors. Type 1 leads to a false positive, leads to a deployment of resources and a lot of money being spent when there's actually nothing to respond to. Whereas type 2, you do not do anything, and the actual biosecurity threat grows, the area infected grows, and you actually end up with a bigger problem. Both of those ultimately result in a lack of confidence in the diagnostic and, to some extent, the entire biosecurity system. So myrtle rust is very easily diagnosed. It's got those bright yellow spores on leaves erupting from pustules, and it's not just on leaves, sometimes on stems, on flowers. But an absence of symptoms does not mean an absence of a pathogen. And Rob Beresford and colleagues have shown that how important temperature is in the infection cycle of Ostropyxina on plants, and in particular long-term and latent infections when it gets below about 8 degrees, which means the plant is infected but showing no symptomology. And also the prepustule lesions on leaves and stems are not definitive. They could be caused by anything. It's not until you get those bright yellow spores that you know that you've got myrtle rust. And also there are hidden infections inside fruit and seed capsules with this in this pathosystem. So Ostropyxina is an invasive pathogen. So this is a graph from Angus and Jeff's review paper of the incursion response in Australia a couple of years ago. And you'll see just before 2020, basically the line's almost going vertical, and most of those invasions are due to the pandemic biotype, the one that we've got here in New Zealand. Also, Ostropyxina is an evolving invasive pathogen. So a graph here from Jane Stewart's paper from a few years ago, looking at the genetic clustering of the different types of Ostropyxina from around the world. Now, this one does not include the South African biotype, and it identified 9 genetic clusters, and the pandemic is made up of the red and the green, so that's C1 and C4 actually comprise what we define as the pandemic biotype. So we already know that there's genetic variability in this pathogen. So we need to have a diagnostic that's differential. We know there are at least 8 genetic variants of Ostropyxina. So there's 3 biotypes, 4 genetic clusters, and the South African genotype. So they're genetically distinct, and 7 of them that we don't have here are genetically distinct from the pandemic biotype that is already present here. Now, we know from work that we've done and others have done that there is limited resistance to the pandemic biotype in Aotearoa. And generally speaking, in invasion biology, the introduction of additional pathogen genetics can break resistance that's currently present, and we do not want that to occur. Further, what we've done and others have done, we already know that the South African genotype and a South American eucalyptus isolate can infect Aotearoa New Zealand species. Now, we do not know whether the basis of resistance susceptibility for those 2 is the same as the pandemic. That's work that we've still got to do. So what we're looking to do is develop a diagnostic test that can detect and identify non-pandemic variants of APSI to ensure that we keep our borders secure. So this research is part of an international collaboration. So we've got New Caledonia involved in our research down here in the Pacific, but this is part of a worldwide network led by Jane out of the University of Colorado. And the initial work that was done in Colorado was to select some putative differential regions from the available genomes Rob talked about the high quality genome we produced for the pandemic. There are also genomic data available for the Eucalyptus guava and South African genomes. And our initial assessment was against Aotearoa pandemic DNA preps, so from isolated from infected plants here. And we're choosing to use a colorimetric amplification assay. In this case, this is a LAMP. It's not PCR. And and red is a control. It's a negative, as it should be. Yellow is a positive, and orange, we're not certain. So right now we're having a little bit of trouble about translating the diagnostics that was developed overseas into our situation. So we've still got some work to do. We need to reassess our positive control. So that's a synthetic positive control because we can't bring those organisms into the country to act as positive controls. We also need to investigate some more of the isothermal parameters, so the temperatures that we use, and also the effective pH. We know that pH can cause type 1 aerosol false positives in these type of assays. And then there are other isothermal platforms to explore. So is LAMP the best option? There's lots of other technologies available. And thank you very much for your attention.
Thanks, Grant. Another really interesting session. Look, we've got a panel discussion now. So can I introduce all the speakers you've heard? We've got Grant, of course, Eiren, we've got Nick, we've got Haami, and we've also got Dave Milner and Marion Wood, who've also been working in the space. So we've got a few questions, I think, that are in the chat, and one of them that I've seen was from maybe we can do this bit of a tag team here, from Bill Dick, asking just around the what were the key chemicals found in your analysis, and has there been any work done on applying them to the forest floor? I think it was to Eiren. Is that right, Bill? Well, I'm just trying to find the question now. It's gone from my
I saw that question pop up. I think it's in our present chat as well.
Can you answer that one? Yep.
Sure. So the flavonoids from Kānuka that we published back in 2019, they're some of our most promising results, but we've also got some other extracts and compounds that seem really promising at the moment, but we have to progress those, as I said, through plant trials, which will be happening this year before they can be used out in the field and before we can be confident that they will work effectively as we want them to.
Great, that's good. Now we've got another question here from Monique Hall asking just a general question to anyone. Well, that's nice and broad around her research isn't in the Kāori space, and I was wondering about the general feeling. Let's get this. What's the general feeling? Bear with me. Is about whether we are making any positive advances in saving our Kāori from extinction. Does it feel like doom and gloom, or is there still hope? Is there still some hope? Yeah, so I'm an optimist. I think there's a lot of hope here, but look, I'll leave it to some of the experts to give us their view on this. Yeah, this is what this conference is all about. Yeah, the work we're doing to actually turn this tide around. But over to who wants to put the hand up to answer that? Nick, you're well placed. Haami.
So, everything I've heard today has really been locked right in, interlocked with the things I was saying. And in fact, I'm probably running behind time in terms of what's happening, and I'm really excited by it. And I think for the first time, we may be on the threshold of finding some real solutions. And I think it's fantastic.
Hey, I'm very supportive of your view as well, Haami. And who was putting the hand up then from the panel?
Good. Rob, thank you.
Dave, yep, there you go.
And tautoko matua Haami's kōrero. And, the challenge or the weta is still out there to save one kauri from one of our kaumatua.
Yes.
And so we're still yet to do that. However, it's really talking about shifting the kōrero from, talking about kauri dieback to talking about kauri ora. So there's simple things like that we can shift the whole paradigm of thought and process towards that. And so, although we haven't, so that shifts the things from the comments around, is it a doom and gloom scenario or situation? I guess it has been, but we're shifting that thought process and the kaupapa towards a holistic approach and really talking about kauri ora now rather than kauri dieback. So if we go in with a clean thought and āhua into our ngāhere, going from kauri dieback to kauri ora, then that whole character and āhua of the ngāhere feels that, and our karakia supports that. So we're shifting the paradigm.
Cool. Sure. Good. Anyone else want to add anything to that, Nick or Grant or Maureen? I think that, yeah. And look, I think Haami, I think we're catching up to you. You're not catching up to us. I think we're catching up to you. Just saying. Shall we move to the next?
Oh,
yeah.
Kira, I would just like to say that also what you've seen today is just a snapshot of some of the work that's being done. There is a whole plethora of work that's out there, screening for trees that are more tolerant to the disease as well. We've got, New Zealand's got nurseries full of trees that hopefully can be replanted that, will be able to sustain their time through having the pathogen present in the environment. So, don't take this as a be all and end all. Do look for the other research that's out there. Kia ora.
Yeah. Sure, thank you. Thanks. Now, did you see that question, Eiren, around do the compounds you're looking at leach from growing trees, either roots or leaves, or do you need to extract them?
They are extracted from the roots and leaves of the Rarawa plants. So yeah, that's the idea, that they're naturally present in the soil and beneficial for the forest. So if we can isolate the actual compound inside these roots and leaves that's really effective against Phytophthora agathodicida, then it will be the safest and effective treatment for kauri dieback that we can then put back into the soil and be confident that it's not going to harm any other organisms, beneficial organisms that are present, or any other plant life around. So, yeah.
Cool. Thank you. Thanks, Sarah. Now, one for you, Grant, around you highlighted the need to develop tools, but also just making the point around it's one thing to develop the tools, but actually implement them. Are you confident? I guess I think that just the question is, the tools that are being developed can be implemented for biosecurity type purposes.
Yes, we've developed isothermal detection technologies for other pathogens, so bacteria, and successfully use those tools basically off the back of a ute on the side of a paddock. So, and the whole reason that we're going down an isothermal route is it's a lot easier to take kits into locations, whether it be a paddock or a forest, and take samples and get immediate results there and then. It's a lot better than having to take samples back to a reference lab where you have to run PCR or something. You get results in 20 minutes in the field with these isothermal technologies.
And that's where we'll get the impact. So it's great to hear, Grant.
Well, if you've got a result, then you can make a decision about what to do there and then. You don't have to wait 24 hours whilst waiting for a diagnostic result.
Yeah, great. Anyone see any other questions or any other comments the panel would like to make before Nick wraps up? Yep. I think
I think that's it. Thank you, Rob, for chairing this wonderful session. Kia ora to all our speakers, matua Haami, Eiren, Grant, and of course our wonderful theme leads, co-leads there. It's been it's a journey of discovery, and really I think the strength is bringing all knowledges, all types of science, knowledge, research, practitioner, whether scientist, academic, on the ground, kaitiaki, are all working together. I think that's the optimism I have. So I think we have to banish doom and gloom, banish dieback, and go on the Waka together and for ourselves and our biodiversity. So kia ora. And another virtual hall Paki to all our speakers, and of course Rob, our wonderful chair today. So kia ora. And as an MC duty, I have to remind you to go back to the program, because we've got 3 more sessions today, and you can see what the next one is through our program. But just encouraging you to stay online, see what's out there, go to our sponsors, go to the discussion rupu. This is quite an amazing platform. So, and we're going to go over to Holden for karakia to close us for this session. Kia ora, Holden.
Oh, kia ora, Nick. Thank you for that. It really resonated. Before I do close the session for this morning, or for this part of the session, something that really resonated for me was something Haami said around, 2 arms are better than one. And that's, to my mind, that's what it is all about here. That's around the 2 treaty partners, Te Krauna, me ngā whānau hapu me ngā iwi, working together around some of these wicked problems. So kia ora Haami, o tira kia ora koutou katoa, ka nui te mihi ki a koutou. Hoi anō hei whakakapi I tēnei wāhanga, ana ka takina he paku karakia, kia oti pai ai tā tātou mahi I tēnei ata. Nā reira kia taunga manaakitanga te mea ngaro, ki runga ki tēnā o tātou. Kia mahea te hua mā kihi, kia toi te kupu, toi te mana, toi te aroha, toi te reo Maori, kia tūturu ka whakamaua, kia tīnga. Hui e tā iki e, kia ora tātou. Kia ora.